All right, are there any questions? So I returned the exam number 1. Okay, most people did very well. You know, there were maybe 4 or 5 people that didn't do so well. Okay, but other than that, most people got like almost 90% or more. Okay, so which is good because I mean, how many people here have had a prior biological background, right? Okay, so only 1 person. So that means I did a pretty good job. You know, people learned something. Okay, although people, somebody told me there were 300 pages and all that. Well, I mean, you're supposed to learn the molecular biology in 6 weeks. You know, you're an engineer. You don't have, you know, semesters or years to learn the stuff. So, you know. And we are getting pretty close to the end of the molecular biology portion. So we'll switch to the engineering thing probably from, yes, probably from sometime next week. And then towards the end of the semester, you know, we need to leave at least a couple of weeks, right? Because you guys are going to make presentations, all right? You're going to present some papers, okay? Once you finish the molecular biology, then you look at the different techniques, all right? The signal processing techniques, then you pick your paper and you'll present. So we have 17 people here, so we need at least 2 weeks, okay, or more than that. So we'll get done, we have to get done well before Thanksgiving because, you know, December 3rd is when I want to finish up the, you know, the last class, okay, December 3rd, right? So, all right, are there any questions? So last time we were, you know, getting ready to take a look at mitosis, right? And towards the end of the class, I mentioned that although mitosis proceeds as a continuous sequence of events, it is traditionally divided into 5 stages: prophase, prometaphase, metaphase, anaphase, and telophase, right? And on your next exam, you can expect that we will ask you to elaborate on these, okay? So this picture is a typical pair of sister chromatids. The chromosomes have replicated, right? They are attached near their center, right? And then there is this protein that is called a kinetochore. So this region is called the centromere, right? We talked about telomeres, centromeres, and replication regions, okay? This centromere actually attracts the protein that is called kinetochore, right? And this kinetochore is useful for binding the microtubule, right? That actually is going to pull the 2 chromosomes apart. Right. And we'll look at the detailed steps in a few minutes. So the different stages in mitosis are as follows. The first one we said was prophase, then prometaphase, metaphase, anaphase, and telophase. Right. So during prophase, the replicated chromosomes condense and the mitotic spindle begins to assemble outside the nucleus. Then during prometaphase, the nuclear envelope will break down, allowing the spindle microtubules to contact the chromosomes and bind to them. Then during the next phase, that is called metaphase, the mitotic spindle gathers all of the chromosomes to the center equator of the spindle. We will elaborate on each of these phases in a few minutes, okay? So don't worry, I mean, this is just a 1-sentence description. Then during anaphase, the 2 sister chromatids in each replicated chromosome synchronously split apart and the spindle draws them to the opposite poles of the cells. And the last step of mitosis is telophase, during which a nuclear envelope reassembles around each of the 2 sets of separated chromosomes to form 2 nuclei. Now in 3 of the stages above, all right, we have talked about the mitotic spindle, all right? See spindle, all right? So in 3 of the stages, then microtubules, all right? Then this mitotic spindle, right? So a natural question that comes up is how is the mitotic spindle formed and what exactly is its role in the cell? Now the mitotic spindle will start to assemble in prophase, right? That means just at the beginning of mitosis, even when the nuclear envelope is intact, the mitotic spindle will start forming, right? Now towards the end of the S phase, again, what is the S phase? What does S stand for? Synthesis, yeah, DNA replication. So towards the end of the S phase, right, the cell is going to duplicate a structure that is called the centrosome to produce 2 daughter centrosomes, which initially remain together at one side of the nucleus, right? As prophase begins, the 2 daughter centrosomes separate and move to the opposite poles of the cell, driven by centrosome associated. And again, I mean, if things have to move inside cells, you need energy, right? You're going to need to use up ATP, right? So driven by centrosome associated motor proteins, right? Remember, we talked about motor proteins in the beginning, the things that can walk, all right? So motor proteins that use the energy of ATP hydrolysis to move along the microtubules, right? So each centrosome will serve to organize its own array of microtubules, and the two sets of microtubules can then interact to form what is called the meiotic spindle. And I'll show that to you in a picture. The next figure shows the two centrosomes with their own array of microtubules. Right? So let's look at the picture. So here, so at some point, all right, the cell duplicates the centrosomes, all right, and you have one centrosome over here, another one here. These chromosomes are inside the nuclear envelope, all right, initially to start with. Now the way, you know, these microtubules are formed is that, or even the meiotic spindle is formed, is that there is, each of these centrosomes, okay, it is going to grow something, grow and shrink, right? So there are units of a protein, right, called tubulin that are added on, and then, you know, radiating from each microtubule, each centrosome, all right, you will have these guys, all right, it's like they're exploring the interior of the cell, growing, shrinking, right? It's a dynamic instability, right? It's like, you know, if things have to go from, let's say, from here to Houston, we have the highway laid out, okay, but when the cell needs stuff, it is going to lay out the highway as it needs it, okay, and then you know, wind it back up once the job is done, okay. So during pro-metaphase, these centrosomes have moved towards two ends of the dividing cell, okay, and they are called the poles, okay. These are called the poles. So if these are the poles, then this line will be the equator, right? I mean, okay. So now, so what is going to happen is that this one, the nuclear envelope is still intact. This the microtubules cannot contact these, you know, the sister chromatids inside. But this one is continuously growing and shrinking, right? It's producing this array of microtubules. Now, while the nuclear envelope is still intact, right, it may happen that some of the microtubules that are growing from this one and the ones growing from this one, they come in contact, okay? And then they are stabilized against disassembly, right? And they form what are called polar microtubules because they link together the two poles, okay? These two are called the poles, right? The spindle poles, right? So that's what's going to happen in the beginning. Now later on, so this is a prophase, all right? Then during pro-metaphase, the nuclear envelope will disintegrate, right? So this was enclosed inside a nuclear envelope. None of these microtubules could reach it. But then in pro-metaphase, the nuclear envelope just, you know, falls apart. And then some of the microtubules will be able to come and link up with the kinetochore proteins, all right, in the centromere region, right? And this would be called, this kind of a microtubule will be called a kinetochore microtubule, all right. It plays the important role that, you know, when the two chromosomes have to be separated, all right, this is the thing that will pull it, pull one copy this way, another copy that way, okay. So that, and there are a number of other things that happen when, you know, like when the two chromosomes move in two different directions, okay, it is because of a number of things. First, this guy shortens, all right, these two guys, they will shorten, all right, because by loss of tubulin units, right, they will shorten, all right, number one. So that will pull this apart. At the same time, these poles can also move away from each other. How? If there is more growth over here, okay, at this end, if more tubulin units are added, then this thing can come apart, right? So that's another, and we are going to talk about each of these in detail, okay? They're called anaphase A, anaphase B, when that happens later, right? So in this picture, we are showing the polar microtubules which are attached to the, which are basically linking the two spindle poles, all right? Then we are also showing the kinetochore microtubules, which can only come into existence once the nuclear envelope has disintegrated. And also there will be microtubules that don't link up. You know, it starts from here, keeps going, coming back, may not have linked up to anything. These will be called unattached microtubules, right? So there are three categories of microtubules here. So this is basically, you know, textual explanation of the stuff that I showed you in the figure, but let's just go through it. So the microtubules that radiate from the centrosome in an interphase cell will continuously polymerize and depolymerize by the addition and loss of units of a protein that is called tubulin, right? So individual microtubules therefore alternate between growing and shrinking, and this process is called dynamic instability, right? It's growing, then shrinking, growing, shrinking. Now, the rapidly growing and shrinking microtubules extend in all directions from the two centrosomes, exploring the interior of the cell, right? Now, during prophase, while the nuclear envelope is still intact, it is possible that some of these microtubules could come in contact with each other from the two spindle poles, right? And so some of these microtubules become stabilized against disassembly to form the highly organized mitotic spindle, like I showed you in that picture. Right? So this is, you're referring to this part, right, where the two have come in contact with each other and now they are not going to shrink anymore. Right? So this happens when some of the microtubules growing from opposite centrosomes interact, binding the two sets of microtubules together to form the basic framework of the mitotic spindle. Right? And those interacting microtubules are called polar microtubules, like I showed you in the picture, since they originate from the two poles of the spindle, and the two centrosomes that give rise to them are called the spindle poles. Okay, so if those two are the poles, all right, you agree with me that this is the equator, right? North pole, south pole equator, right? Because that terminology will be useful in a few minutes. So these are the spindle poles, right? Now, chromosomes will get attached to the mitotic spindle during prometaphase, right? So prometaphase starts abruptly with the disintegration of the nuclear envelope, right? And following this, the spindle microtubules, which have been lying in wait outside the nucleus, suddenly will gain access to the replicated chromosomes and will bind to them, right? The spindle microtubules bind the chromosomes through specialized protein complexes, which are called kinetochores, which are formed on the chromosomes during late prophase. Okay. We talked about the centromere region and the kinetochore coming in and binding there. So as we already mentioned, each replicated chromosome consists of two sister chromatids joined together along their length, and each chromatid is constricted at a region of specialized DNA sequence which is called the centromere. Right? And just before prometaphase, kinetochore proteins will assemble into a large complex on each centromere. So it gets those chromatids ready for binding to those microtubules. So once the nuclear envelope has disintegrated, a randomly pointing microtubule encountering a kinetochore will bind to it, thereby capturing the chromosome, right? Like I showed in the figure. So such a microtubule is called a kinetochore microtubule, and it links the chromosome to a spindle pole. Now, as I said before, not every microtubule emanating from the two centrosomes will end up forming a kinetochore microtubule or a polar microtubule, right? In fact, several of the microtubules will remain unattached, and these are called unattached microtubules. Now, during prometaphase, the chromosomes which are now assembled to the mitotic spindle, right, they will appear to move as if jerked around randomly in different directions, right? So they are being aligned along the equator. So finally they align at the equator. That's why I said those two are the poles, the line, perpendicular line in the middle is the equator, right? So finally they will align at the equator of the spindle, halfway between the two spindle poles, thereby forming the metaphase plate, right? And once that happens, once they are lined up at the metaphase plate, that basically signals the start of metaphase, right? Before that, it's called prometaphase. So the formation of the metaphase plate defines the beginning of metaphase, right? Now the precise forces that act to bring the chromosomes to the equator are not well understood, okay? How they are pushed around this way, that way, and then lined up, not well understood, you know, but maybe, again, my notes are like 10 years old, so maybe today it might be well understood. I don't know, okay? Yeah, just today somebody got the Nobel Prize for medicine for, you know, figuring out the GPS kind of stuff in the brain, you know. So, I mean, a lot of things have probably been happening in the meantime. So. Now, however, the continual growth and shrinkage of the microtubules and the action of microtubule motor proteins are thought to play a role. Right. Now, in this context, see, this is a dynamic thing. These microtubules have to be dynamically maintained, right? Like they're growing, shrinking, growing, shrinking. If you go and mess up with the growth process, all right, then the cell cannot divide, right? So in this context, we know that a continuous balanced addition and loss of tubulin subunits is required to maintain the mitotic spindle. So thus, when tubulin addition is blocked in a mitotic cell, using, say, the drug colchicine, tubulin loss continues until the spindle disappears. See, the cell is trying to divide, okay? If you go and mess up the spindle, the chromosomes cannot be separated, all right? And the cell cannot divide. And this is used in cancer therapy. You know, they have drugs that are called antimitotics, okay? So if you give that drug, it is going to basically prevent the cell from dividing, right? So this prevents the cell from dividing, and so colchicine can be used in cancer therapy to prevent the cancer cell from proliferating. The only problem is that they don't know how to prevent the normal cells from not doing the same thing, okay? So if you hit somebody with colchicine, even their normal cells also will die, okay, will not divide. So we have talked about prophase, metaphase, metaphase, and then the next phase in mitosis is called anaphase. So at the start of anaphase, the connections between the two sister chromatids are cut by proteolytic enzymes. Again, proteolytic means enzymes that break down proteins, all right? Allowing each chromatid, which is now called a daughter chromosome, to be gradually pulled to the spindle pole to which it is attached, right? And as I said before, this movement is happening because of two things. The kinetochore microtubules are shortening, right? And then also the poles are moving away from each other, right? So this movement is the result of two independent processes brought about by different parts of the mitotic spindle. These processes are called anaphase A and anaphase B, and their occurrence is more or less simultaneous, right? In anaphase A, the kinetochore microtubules shorten by depolymerization, by losing tubulin units, and the two daughter chromosomes move towards their respective spindle poles. In anaphase B, the spindle poles themselves move away from each other, further contributing to the segregation of the two groups of daughter chromosomes, right? And the driving force for the movements of anaphase A is thought to be provided partly by the action of microtubule motor, and that means movement generating proteins operating at the kinetochore, and partly by the loss of tubulin units, subunits that occurs again mainly at the kinetochore end of the kinetochore microtubules. Right? So it will lose some tubulin units, then there will be some movement also due to some proteins, and then they will move apart. Right? Now the driving force for the moving apart of the spindle poles in anaphase B is thought to be provided by the polymerization of the polar microtubules by the addition of tubulin units at their free ends. Like if you have, see if you have more, let's go back to this picture. If you have more of these units getting added, then the thing will move, the two poles will move apart. Right, more units added over here. That means the common region, if it is made longer, you know, so, or yeah, the common region will become shorter with the addition of more units here. Now the last step in mitosis is telophase, okay? Because now the two chromosomes have separated out, and now you need a fresh nuclear envelope to be formed around each daughter chromosome. So during telophase, the nuclear envelope reforms around each group of chromosomes to form the two daughter nuclei. Now with the creation of the two daughter nuclei, the process of nuclear division, which is mitosis, is complete, right? Now M phase involves more than just the segregation of the daughter chromosomes and the formation of new nuclei, because everything else also has to be properly apportioned between the two daughter cells. Okay? So it is also the time during which other components of the cell, such as membranes, organelles, and proteins, are distributed more or less evenly between the two daughter cells. And this is achieved by what is called cytokinesis, or splitting of the cell, and usually begins in anaphase, right? That means when the chromosomes are moving apart, but is not completed until after the two daughter nuclei have formed. Now regarding cytokinesis, we note the following. The mitotic spindle will determine the plane of cytoplasmic cleavage, right, which is usually orthogonal to the orientation of the mitotic spindle. So you have the mitotic spindle that is going to pull the chromosomes apart, right? The splitting of the cell will take place perpendicular to it. And people have done tests where, you know, if you use a needle to disturb the mitotic spindle, then the plane of cytoplasmic cleavage also will shift accordingly, right? If it's early enough, of course. Right? Now the thing that carries out this cytokinesis is called the contractile ring of animal cells, and it is made up of proteins actin and myosin. Now in the case of plant cells, right, the cytokinesis or the splitting of actual splitting of the cell involves the formation of a new cell wall, presumably because the plant cells in general are surrounded by a tough cell wall. So this completes our discussion of mitosis, right? So next, so mitosis is for somatic cells, that is the cells in the body, okay? Meiosis is basically for the reproductive cells. So how, starting from diploid cells, that means cells which have 2 copies of each chromosome, you end up with the gametes, the egg and the sperm, which have only 1 copy of each chromosome, right? So we have already taken a look at meiosis, all right, in the context of what? Anybody remembers? I think we talked a little bit about meiosis before, right? I mean, when was that? Why did we talk about it? Yeah, go ahead.
When go from one direction to the other, and what's it called? Natural way.
Yeah, genetic variation. Yeah. In that chapter we talked about it to show that how much of variation can arise because of sexual reproduction, right? So in this section, we are going to revisit meiosis in a little bit more detail, right, and point out the important differences between meiosis and mitosis, right. So this is a kind of cell division employed by sexually producing diploid organisms to produce the reproductive cells. Now, in a diploid organism, all the cells of the body, including the germline cells that give rise to the gametes, are diploid. However, the gametes themselves are haploid. That means they have one copy of each chromosome. Thus, the haploid gametes must be produced from their diploid precursors by a special type of cell division, and this special type of cell division is called meiosis. Now, with the exception of the chromosomes that determine sex, the sex chromosomes, right, that is the XX and the XY chromosomes, a diploid nucleus contains two very similar versions of each chromosome. One from the father, which is called the paternal chromosome, and one from the mother, that is called the maternal chromosome. The two versions of each chromosome, however, are not genetically identical, as they carry different variants of many of the genes. They are both functional. In normal people, they are both functional, but they're a little bit different. They are therefore called homologous chromosomes or homologs, meaning that they are similar but not identical. A diploid cell in a sexually reproducing organism consequently carries two similar sets of genetic information. In most cells, the paternal and the maternal homologs maintain a completely separate existence as independent chromosomes, right? It's like they don't know each other, right? However, during meiosis, they come and line up, right? So mitosis and meiosis are similar in certain respects, but there are important differences that we'll be discussing. The following figure illustrates the key differences between mitosis and meiosis using a fictitious diploid cell with only one pair of homologous chromosomes. So this is the picture that we have, right? So if you have mitosis, all right, so, and again, I mean, we are just showing one pair. There, in a human cell, there are 23 pairs of these. Okay, so let's say the red one is the maternal homolog, the black one is the paternal homolog. So if this cell is going to undergo mitotic cell division, right, the DNA will replicate, right? So now one copy of each chromosome has to be given to each dividing cell. You know, so after the DNA has replicated, right, then you will have, you know, this is the mitotic spindle, the spindle poles, all right, over here. Then these guys are going to get pulled, all right, so for the one cell splits in two, and then each one gets a copy of the paternal chromosome and the maternal chromosome, right? So chromosomes that each cell inherits, they are, you know, barring of course errors during DNA replication, they are identical. Okay, so that's what happens in mitosis. Now, in the case of meiosis, all right, see, really, you know, meiosis could have happened like this, because this has two copies of each chromosome, all right? You want to produce gametes that have only one copy, okay? So if you skip the DNA replication phase, all right, and just split the cell in two, all right, sending one chromosome to each cell, all right, that's how meiosis could have worked, okay? But for some unknown reason, that's not how it works. Instead, there is DNA replication, all right? That means instead of one round of cell division to produce haploid gametes, you're now going to need 2 rounds of cell division. So there's first DNA replication on both the paternal and the maternal homolog, all right? And then the funny thing is that these two guys, okay, before this, it's like they don't know each other. Now, let's say for chromosome number 1, the paternal and maternal homolog, they will line up next to each other, okay? Because they're homologous sequences, all right, they're homologous chromosomes, there will be some crossover, all right? Some pieces of one will go into the other, right? And after that, you will need 2 rounds of cell splitting, right? So once the cell divides, you still have 2 chromosomes in each one of them, okay? But again, notice there has been crossover. Portion of the black one is now in here, portion of the red one is in the one on the right, okay? So 1 round of cell division, then you need another round of cell division to produce the haploid gametes. So you do that, and now you have 4 different possible chromosomes, right? See, because of the crossover. And this is what we were referring to earlier, that there was going to be a lot of genetic variation because of this. So mitosis and meiosis are similar in certain respects, but there are important differences that we'll be discussing. In mitosis, each chromosome replicates and the replicated chromosomes line up in random order at the metaphase plate, right? The 2 sister chromatids then separate from each other to become individual chromosomes, and the 2 daughter cells produced by cytokinesis inherit a copy of each paternal chromosome and a copy of each maternal chromosome, right? Thus, both sets of genetic information are transmitted intact to the 2 daughter cells, which are therefore each diploid and genetically identical. Now, in contrast, when diploid cells divide by meiosis, they form haploid gametes with only 1/2 the original number of chromosomes. That is only one chromosome of each type instead of a pair of homologs of each type. Thus, each gamete acquires either the maternal copy or the paternal copy of a chromosome, but not both. Right? This reduction is needed so that when two gametes of opposite types, an egg and a sperm in animals, fuse at fertilization, the chromosome number is restored in the embryo to the original diploid number for that species. Right? So the embryo, if it's a human embryo, then it should have 23 pairs of chromosomes. Right? So that's why you have this halving. Now, since the assignment of maternal and paternal chromosomes to the gametes during meiosis occurs at random, the original maternal and paternal chromosomes are reshuffled into an incredibly large number of different combinations, as we discussed earlier in the chapter on genetic variation. So as noted in that chapter and as shown in the figure, in a meiotic cell division, the replicated homologous paternal and maternal chromosomes, including the two replicated sex chromosomes, pair up alongside each other before they line up on the spindle. Right? And this makes it possible for crossovers by homologous recombination to occur at this stage. Now, in principle, since meiosis involves the halving of the number of chromosomes, it could have occurred by a simple modification of a normal mitotic cell division, such that the DNA replication, S phase is omitted. If you omit the S phase, you do a cell division, right, you're going to be halving the total amount of DNA, the number of chromosomes, right? So for unknown reasons, that doesn't happen. The actual meiotic process is more complicated and involves DNA replication followed by two cell divisions instead of one, as shown in the earlier figure. Now, occasionally, see, when you're having all this division, okay, when you're having this division, right, the meiotic division, and then each cell is supposed to get one copy of each chromosome, right? What happens when this guy divides if the same cell got two copies, okay, and the other one got nothing? The other one will be eliminated because it is not viable, but this one will have two copies, okay? And you can have situations like, one example is the Down syndrome, all right? It's called trisomy 21 because the individual that has Down syndrome has got 3 copies of chromosome 21, okay, instead of 2 copies, okay? So probably it's either an egg that had 2 copies of that fertilized by a sperm with 1 copy, all right, or vice versa, okay? But in most cases, those individuals do not survive, right? Another example is if this involves the sex chromosomes, all right, because females will have 2 X chromosomes, all right? Males will have X and Y, okay? What happens if somebody has 2 X chromosomes and 1 Y chromosome, right? That person will, no, it happens. That person will have both male and female characteristics, all right? It can happen. So it's not legend, or it's not, you know, something that just people talk about, you know, it can happen, right? Or somebody could have 3 X's, okay? So triplo X, all right? So, I mean, most of the time it doesn't happen, but if this thing makes a mistake, it can be quite disastrous. Yes, sir. I didn't understand. If chromosomes are what? Oh, twice mRNA it will produce. Is that what you're saying? Yeah, probably, yeah. No, if you have an extra copy of a gene or something, yeah, maybe your cells will overexpress that thing, okay? And in most cases, it will be lethal because that person will not survive. But, you know, Down syndrome is an example, okay, that you have one extra copy of chromosome 21, right? And if you've seen people with Down syndrome, I'm sure most of you have seen, okay? I mean, once you see, I mean, you can make out that there is something abnormal about them, you know, so the way they carry themselves and all that. So you can make out, you know. But in most cases, you know, if the chromosome number is not correct, right, then that individual will not survive. You know, they will not even. I mean, that kind of a fetus will probably not even make it to full term. You know, so will automatically get eliminated from the population. But in the few cases that they do, right, and these are abnormalities that they can detect. You know, they do all this test that is called amniocentesis, where they'll take amniotic fluid. And remember, I talked about karyotyping, finding out the different chromosomes and all that. You know, you can figure out how many chromosomes there are. You know, so and it is possible, you know, to predict in advance, you know, what is going to happen to that particular individual, right? But then, of course, it brings up all kinds of ethical issues. You know, what are you going to do, right? If it's going to be somebody with Down syndrome or something else, it's a decision that the parents and the doctors have to make, and so there are ethical issues that come up. So occasionally, as I said, the meiotic process occurs abnormally and homologs fail to separate. This is a phenomenon that is known as non-disjunction. All right. In this case, some of the gametes that are produced lack a particular chromosome, while others have more than one copy of it. Such gametes, when combined with a normal gamete of the opposite type, right, it's very unlikely that you're going to have, a gamete that has extra copies of chromosomes, again combining with another set of gametes that have extra copies. That probably won't happen. So it probably will combine with a normal gamete. They will form abnormal embryos, most of which do not survive. Some, however, do, and Down syndrome in humans is an example of a disease that is caused by an extra copy of chromosome 21. And this condition is therefore referred to in the scientific literature as trisomy 21, meaning that such a person's genome has 3 copies of chromosome 21 instead of the 2 copies that normal humans have. All right, are there any questions? So we finished the chapter on cell division, right? So now we are going to move on to the chapter about cell cycle control, cell death, and cancer, right? So in this chapter, we introduce the phenomena of cell cycle control and programmed cell death in multicellular organisms and discuss how disruption in either of these can lead to the disease called cancer, right? See, in multicellular organisms, even for an adult organism, there has to be cell division has to continue. Because as I told you before, the cells on your skin are replaced once every 10 days. The cells lining your gut once every 3 days. So there is some kind of a dynamic equilibrium. The rate at which the new cells are being produced is roughly the rate at which the old cells are being sloughed off, right? So there is a dynamic equilibrium. If that is disrupted, then that's when you can get diseases such as cancer, right? Now, our discussion so far has focused on 2 aspects of cell division: replication of the contents of the cell, right? And we talked about that, like you can do DNA replication, protein synthesis, and so on. That's how you replicate. Then in the last chapter, we talked about the actual partitioning of these replicated contents between the 2 daughter cells when the cell divides. Okay, how does it take place? All this mitotic spindle, prophase, prometaphase, anaphase, telophase, all that stuff. Okay. Now there is yet another aspect of cell division which is crucially important, namely the mechanism by which the cell controls the different chronological steps that are involved in cell division. Because you have like S phase, all right, then G2 phase, all right, M phase, G1 phase, and so on. Okay, so one step shouldn't happen before the other one is completed. So there has to be some kind of clock that is doing the timing, right? The timing is important. So the events of the cell cycle occur in a fixed sequence, namely the M phase. So the M phase is when mitosis occurs, mitosis and cytokinesis, all right, followed by the G1 phase. Again, G1 is for what? Gap, yeah, gap 1 phase. G1 phase followed by S phase, then S phase followed by G2 phase, and finally G2 phase followed by M phase again. So this is ensured by a cell cycle control system which has to perform a number of functions. First of all, the cell cycle control system has to activate the enzymes and the other proteins that are responsible for carrying out each process in the cell cycle at the appropriate time, and then it has to deactivate them once the process is completed. Right. It must also ensure that each stage of the cell cycle is completed before the next one is begun. For example, it has to make sure that all of the DNA has been replicated before you trigger mitosis, right? That mitosis has been finished before you do cytokinesis or the actual splitting of the of the cell in 2, and so on. Now, the control system must also take into account if the conditions outside the cell are conducive for division. For example, in a multicellular organism, the control system must be responsive to signals from other cells, such as those that stimulate cell division when more cells are needed. If you have a cut or a wound, okay, you need more cells. Okay, so there will be platelets will give out certain signals to the surviving cells telling them to divide. Okay. So it is clear that the cell cycle control system plays a major role in the regulation of cell numbers in the tissues of the body. Right? And when this system malfunctions, it can result in cancer. Right? If it's a unicellular organism like bacteria or yeast, right, you give them enough food, right temperature, right carbon dioxide concentration, they'll just keep on multiplying. in the case of a multicellular organism, new cells should be produced only at the right time, at the right location when they're needed, okay, not otherwise. And if they happen, in the absence of need, that's when you can get tumors and cancer and so on. Now, this operation of the cell cycle control system is very similar to that of the control system for any cyclic process. And we will illustrate this by drawing the analogy with an automatic washing machine, right? Now, the duty cycle of an automatic washing machine consists of the following 5 steps. Take in water, mix with detergent, wash the clothes, rinse them, spin them dry. Okay, we all know that. We all use washing machines, right? Now, in the eukaryotic cell cycle, these steps would correspond to like DNA replication, mitosis, and so on. Okay, G1 phase and G2 phase and so on. Furthermore, the washing machine controller is itself regulated at certain critical points of the cycle by feedback, right? There's feedback control from the processes that are being performed, okay? For example, if the level of water is not high enough, okay, you shouldn't go to the next step, okay? That kind of thing is there. Right? So, for example, sensors monitor the water levels in a washing machine and send signals back to the controller to prevent the start of the next process before the current one has been completed. Similarly, the events of the cell cycle have to occur in a particular sequence, and this sequence must be preserved even if one of the steps is taking longer than usual. For example, all of the nuclear DNA must be replicated before the nucleus begins to divide, right? And it is crucial for most cells to double in size before dividing in two, as otherwise the cells would get progressively smaller at each cell division, right? So there is a question of timing. If something is taking longer than normal, then there should be some breaks, molecular breaks put on, so that you wait for some time, right? Now, the cell cycle control system achieves all this by means of molecular breaks that can stop the cycle at various checkpoints. Right? Like if there is not enough food in the environment, right, the cell shouldn't divide, right? So the control system in most cells has got checkpoints for cell size, where the cell cycle is halted until the cell has grown to an appropriate size. For example, in G1, right, again, G1 is gap 1, right? It's the phase between M phase and the DNA replication phase, S phase. So in G1, a size checkpoint allows the system to halt and the cell to grow further if necessary before a new round of DNA replication is triggered. Now, cell growth depends on an adequate supply of nutrients and other factors in the extracellular environment. And the G1 checkpoint also allows the cell to check that the environment is favorable for cell proliferation before committing itself to the DNA replication or S phase. Okay? It's going to check. I mean, it's not going to do unnecessary work. It's going to make sure that the conditions are conducive for growth. There is a second size checkpoint which occurs in G2, right? That is after S phase, allowing the system to halt before it triggers mitosis, right? The G2 checkpoint also allows the cell to check that DNA replication is complete before proceeding to mitosis. In fact, another example is if there is DNA damage, okay? You do not want to replicate DNA damage, right? You step out into the sun, your DNA got damaged, right? That needs to be repaired, right? So there are breaks in G1, right? In fact, they call that G1 arrest, right? Where if you zap a cell with a lot of radiation, right? So there has been DNA damage, it shouldn't go into S phase, right? So it'll arrest in the G1 phase, right? And wait until the DNA gets repaired and before it triggers a DNA replication. So there are all kinds of control systems at work. Now the cell cycle control system governs the cell cycle machinery through the phosphorylation of key proteins that initiate or regulate DNA replication, mitosis, and cytokinesis, right? Recall from an earlier chapter that phosphorylation reactions are carried out by enzymes which are called kinases, okay? And what are the enzymes which remove the phosphate groups? Phosphatases, yeah. So kinases and phosphatases are extremely important in the control of eukaryotic cell division, right? Now the protein kinases of the cell cycle control system are present in dividing cells throughout the cell cycle. They are activated, however, only at appropriate times in the cycle, after which they quickly become deactivated again, right? And this is made possible by a second set of protein components of the control system, which are called the cyclins, right? Cyclins have got no enzymatic activity by themselves, but they have to bind to the cell cycle kinases before the kinases can become enzymatically active, right? As a result, the kinases of the cell cycle control system are referred to as CDKs in short, or cyclin-dependent protein kinases, right? Now, cyclins derive their name from the fact that their concentrations vary in a, remember there is some timing involved in the cell, okay, so the concentration will oscillate, right, of the cyclins. So cyclins derive their name from the fact that their concentrations vary in a cyclical fashion during the cell cycle. We will focus, and there are different cyclins and different cyclin-dependent kinases. We will focus only on one particular one. So we will focus on the cyclin-dependent kinase complex that is responsible for driving cells into mitosis, right? And this cyclin-dependent kinase complex is known as the M phase promoting factor, or MPF, because at that time people didn't know the composition, so they didn't know it had a kinase and it had a cyclin, okay? But this is the factor that would promote the cells going into M phase, okay? So they called it an M phase promoting factor. The cyclin-dependent kinase complex that drives cells into M phase was first discovered through studies of cell division in frog eggs, right? The fertilized eggs of many animals are especially well suited for biochemical studies of the cell cycle because number one, they are very large, right? A frog's egg is like a millimeter in size, so that's a pretty big, it's a single cell that is pretty big, right? And if it's a fertilized egg, the first few divisions, all right, these are what are called cleavage divisions, where there is just DNA replication and then mitosis. DNA replication, mitosis, all right? So it happens very fast, right? So the fertilized eggs have that characteristic that they will divide very, very rapidly, right? So you can observe things pretty quickly. So this is due to the fact that they undergo cleavage divisions, that is M phase followed by S phase and then M phase again with the little or no G1 or G2 phases in between. Now, by taking frog eggs at a particular stage of the cell cycle, an extract can be prepared which is representative of that cell cycle stage. Like you break the frog eggs up, all right, and you get a soup, all right? So that has all the proteins that are representative of that stage. Now, the biological activity of such an extract can then be tested by injecting it into a xenopus oocyte, right, which is the immature diploid precursor of the unfertilized frog egg, and observing its effects on cell cycle behavior, right? So the oocyte is, like we said that the egg is haploid, okay? The diploid cell from which the egg is going to be formed, right, by meiosis, that is called the oocyte, right? So the oocyte is still diploid, right? And then from that, by meiosis, you're going to get the haploid eggs, right? Now, the xenopus oocyte is a convenient test system for detecting an activity that drives cells into M phase, as it has completed DNA replication and is arrested just before M phase of the first meiotic division, right? To go from the oocyte, right, to the egg, right, you basically need meiosis. So you're going to have 2 rounds of cell division, right? So the DNA replication, see, it has completed the DNA replication, right? And it is just arrested before the M phase of the first meiotic division, right? So the oocyte is therefore at a stage in the cell cycle that is equivalent to the G2 phase of a mitotic cell cycle. And again, I mean, this thing is, I mean, of interest to know, you know, that in females, okay, the number of eggs that is going to be produced, okay, in a lifetime, that is determined before your birth, okay, because it is always arrested. It is like an oocyte, okay. It has already completed the, the first. It is just before the first meiotic cell division, okay. So it is already predetermined, right? So in such experiments, right, so you're taking the abstract, right, from the fertilized frog egg, right? You're taking that abstract, and then the test system, okay, you want to see the activity of that M phase promoting factor. The test system will be basically that oocyte, because the oocyte has completed DNA replication, and you know, if you give it the M phase promoting factor, it will drive it into something similar to mitosis, but it's only the first division, okay, the first meiotic division, right? So it was, in such experiments, it was found that an abstract from an M phase fertilized egg instantly drives the oocyte into M phase. Whereas if you take cytoplasm from a cleaving egg at other phases of the cell cycle, it does not, right? And when initially discovered, the chemical composition and the mechanism of action of this factor responsible for this activity were unknown, and consequently the factor was simply called the M phase promoting factor, or MPF. Now, MPF activity was found to oscillate dramatically during the course of each cell cycle, as shown in the next figure. It increases, let me show you in the figure, right? So this is the MPF activity, right? It goes up, right, and comes down. This is the M phase. It goes up very quickly, then it comes down slow. It comes down also quickly. No, it comes down slow. Let's see. No, it's going to go up to start the M phase, okay? And then once M phase is over, it's going to come down, then go up, then come down, okay? But it's not the activity of just this factor. It's the activity also of the cyclin that determines the speed at which it will go up and come down. So I'm getting to that in a few minutes, right? So MPF activity was found to oscillate dramatically during the course of each cell cycle as shown in that figure. It increased rapidly just before the start of mitosis, and it also fell rapidly to zero towards the end of mitosis, right? So subsequent studies revealed that MPF contains a single protein kinase, right, which is required for its activity. So by phosphorylating key proteins, so right, a kinase is going to phosphorylate proteins, right? So by phosphorylating key proteins, the kinase causes several phenomena associated with mitosis to occur. For example, with mitosis, you need the condensation of the chromosomes, right? You don't want chromosomes that are spread out all over the place. They have to condense. The nuclear envelope has to disintegrate in pro-metaphase, all right? And then the formation of the mitotic spindle. All those things are triggered by proteins, all right? And the oscillation of the M phase promoting factor is going to control the timing. However, the M phase promoting factor kinase is not capable of acting by itself and has to have a specific cyclin that is bound to it in order to function. And to find out the activity of these cyclins, they did experiments using cleaving clam eggs, right? And that led to the discovery of cyclin. Cyclin was initially identified as a protein whose concentration will rise gradually during interphase and then fell rapidly to zero as the cells went through M phase, repeating this performance in each cell cycle, right, as shown in that figure. So this is like the timing component, right? So this is just oscillating like this, right, this cyclin. Okay, it's like the timer, like the clock on a computer, right? And so the MPF, or the M phase promoting factor, is a protein complex which contains two subunits, a regulatory subunit, which is the cyclin, and a catalytic subunit, which is the mitotic cyclin-dependent kinase, right? And I will continue from here next time. Let's stop here for today. I mean, this is a good place to stop. I'll just repeat a little bit of this stuff next time and then go from here. All right, any questions?